but we'll be continuing some of the work and, the train of thought and we'll be working along the same lines. So I'll be happy to hear your thoughts on all of that. Yeah.
All right. Thanks, Ayush, for introducing me. This is my first talk too, so we're on the same boat. And yeah, I'll get started without further ado. Just a second. Wait, can you guys see my screen?
Yeah.
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 Right. Okay. So as Ayush introduced, there are subtitles on this. Yeah. So my talk is titled Brain Proposes, Spinal Cord Disposes. And I will mainly be talking about results from Anu's lab during her postdoc work, as she mentioned. And she worked on characterizing the organization and functions of cerebellospinal neurons. But before I delve right into that, before I go and start talking about what cerebellospinal neurons are, I want to take a step back and talk about so the essence of today's talk will be founded in motor control and motor learning. So basically, I'll be talking about movement in the broadest sense. And so to start with, I want to talk about why we need to talk about movement at all now. At the end of the day, we've been scientifically researching movement for hundreds of years with classically famous experiments such as Boulogn's mechanism of human physiognomy, where he attached electrodes to the faces of his test subjects and elicited facial movement that in social terms translate into expression, and so much more. We've from everything from reflexes to voluntary movements to the pathways that actually facilitate these movements. We've been through different methods, through anatomical methods, through electrophysiological methods, through EMG, through a lot of different perspectives. For many, many years now, we've actually been researching and studying movement. So why we still here, why we still studying movement, why we still doing this talk. And hopefully, as I finish my introduction, we'll get a clearer view of the essentiality of actually digging deeper through these studies that have been going on for so many years and understanding what's actually going on. So to start with, I'll give a list of things that we know about movement. For example, what are the neural coordinates of movement, right? So we know, for example, let's start with the spinal cord. We know that the spinal cord is both sufficient and necessary. Now, these are very important. If it's sufficient, and the fact that it's sufficient may not necessarily imply that it's necessary, and vice versa. So we know for a fact that the spinal cord is both necessary and sufficient in this from classical works by many people, but very famously by Robert Witt, who mechanically stimulated the spinal cord of decapitated frogs, and he found that the frog could still flex its muscles even though it was spinalized, even though its connection to the brain was lost and it only had a spinal cord. So as you can see, there is actually no head here, so there is no brain, but the stimulation of the spinal cord still elicited movement. So we know this. This is one of the classical experiments that have been done like almost 200, 300 years ago. But this is, so we know that the spinal cord, moving the spinal cord or pressing the spinal cord will elicit movement. But how is this happening? That is a question that we'll touch upon, and neuroscience in general is touching upon constantly through research, even in contemporary times. And so in the spinal, so now that from the 1700s, we know that the spinal cord is necessary and sufficient for movement, but it was until Cahal came up with his brilliant neuroanatomical studies of the spinal cord and the brain that still lays foundations for a lot of work. And followed by Sherrington's analysis of how movement is actually facilitated. So basically, he studied reflexes, and he came up with a theory that motor neurons are the final common pathway of any motor behavior. What that means is basically there can be no movement without motor neurons. That no matter what is happening, no matter what sensory inputs are gotten, or no matter what brain commands are sent, unless there is an existence of motor neurons, I'm sorry for the technical, but it should be motor, regardless of what's happening in the rest of the body, without motor neurons, movement cannot be facilitated. And this is one of the very foundational findings or theories in neuroscience that Sherrington came up with. So this is again something else that we know. These have things like this have now become foundation for further research in the field of movement in neuroscience. These form like the basic elements of how movement is constituted. So now we know that motor neurons facilitate other final element in facilitating movement. But what controls motor neurons? Why do motor neurons behave the way they do? And so basically that brings a question of how does the nervous system, the peripheral nervous system, the central nervous system, orchestrate the actual facilitation of movements that finally the motor neurons go and elicit. So basically what controls motor neurons. And so building on that, what we know is that there are three kinds of neurons. There are sensory neurons, which receive sensory information from the outside world and get it into our bodies, and these sensory inputs, which can be of multiple types. They could be mechanoreceptive inputs. Mechanoreceptive in broad terms are touch, are the inputs of touch. It could be nociceptive inputs, which is pain. It could be proprioceptive inputs, which is basically where muscle is in 3D space, so how much a muscle is flexed. So all of these sensory inputs come together and give us a base understanding of how the world is functioning around us, and that helps us elicit appropriate motor behavior. And this is important because if you think about it, all movement has purpose, and movement is really, really important. Almost everything we do, from actual like conscious movement, like playing something, to social responses, to people, to smiling at someone, all of these happen through very specific, very coordinated movements of the body. And these are facilitated by broadly by these three types of neurons and their very intricate interplay. So these are sensory neurons, and there are different kinds that get in different inputs. And we know that sensory neurons themselves can elicit motor neurons directly, causing what we call reflexes. So they don't. So a very high pain stimulus need not have to go all the way to the brain and then come back down to elicit a movement. We know that if we touch something really hot, there's an instantaneous movement that occurs, and so that happens because sensory neurons can directly elicit input into motor neurons, which can then conduct motor behavior. And this has been proven by showing that spinalized animals, which have been severed from their brains, can also conduct movement, which means that it doesn't really need to go all the way up to the brain to elicit movement. Building on that, we've spinalized animals with parts of their brain or the entire brain removed have been shown to be able to walk on treadmills, have been shown to be able to elicit certain, albeit uncoordinated gait movement behavior, certain locomotory activity, even though parts of their brain or their entire brain are completely missing. So the rhythmic movements can be elicited, locomotor movements can be elicited, and this is something that we know. And then that brings us to the second type of neurons that I listed there, spinal neurons themselves. These are neurons that get information from sensory neurons, they get sensory input, and they transmit these inputs to different parts of the spine and the brain. Now, this is important because classically we think that sensory, yeah.
We just lost, or maybe it was.
Can the rest of you hear me?
Yeah, we can hear you.
Okay.
We can't hear you now, though.
You can't hear me properly? Oh, okay. I'm not sure what to do about it.
Yeah, we can hear you. We can't hear Mal.
Oh, you can't hear Anu. Okay. All right. Yeah. So I think it must be a problem on my side. So then there are spinal neurons, which we used to classically think are just relay messengers that just transmit information from the sensory inputs to the brain. But later research has found that that is not true. Spinal neurons can actually talk amongst themselves. And an example of this are central pattern generators, which are kinds of arrays of interneurons that generate rhythmic locomotory patterns that when we walk, we are not consciously thinking of the length of the steps we take or the pressure we put when we take each step. These things come to us in what in layman's terms we'd say unconsciously. But there is something that is actually coordinating this movement, that's actually being able to translate sensory input from where we are walking, from what's on the road, to how we actually move. And it has been found that spinal neurons themselves have actually a very important role in coordinating this behavior that goes over and beyond just relaying sensory input to the brain. And lastly, there's the descending neurons, the third group of neurons that I spoke about. And these are neurons that bring, in the context of movement, motor command inputs from the brain to the spine and hence to the motor neurons to elicit voluntary movements or other kinds of movements that are conscious. And these how these work is these take in all the sensory input that sensory neurons got from the outside environment that were relayed to the brain, into different regions of the brain, to the motor cortex, to different, it could even be decision-making parts of the brain that are connected to the motor cortex and so on. And then integrate all of this information and then send certain motor commands to elicit appropriate behavior, to elicit specific behavior that is most appropriate. So these are broadly the three kinds of the neurons that help coordinate behavior. And what's important is now we know that there are these three kinds of neurons and information travels between them. But this information is vast and it's of extremely varied quality. So there could be information about how much pressure a foot is putting when walking, or there could be information about how fast we are going when we are walking. All of this information, or like how much wind is blowing towards us, that will also help us, that will also elicit a different kind of movement in us. So all of this different information that all of these neurons are carrying are integrated in parts of the brain. And because of that integration and because of specific behaviors that our brain thinks is appropriate because of the result of these integrations of input, we can actually elicit like voluntary control. I can actually reach for something. If I'm reaching for a bottle, that's a voluntary movement. But a lot of different inputs are coming into this voluntary movement, and all of these inputs are translated into a specific kind of movement. So this is what's important. This is kind of the frontier of what's happening, of how movement control is studied. So how are all these inputs integrated for voluntary movements? Now, as I mentioned, there are multiple different kinds of sensory inputs. There's sight, hearing, pain, touch, and so on. And these are coming from these sensory inputs, and they are integrated in the brain and then sent back to the spinal cord to elicit motor behavior. And this creates a very powerful system of coordination that we honestly don't really think about. We go through daily life constantly moving, but we usually don't think about how specific movements are possible, how we respond to things in the environment to actually behave a certain way in the context of movement. So now, building on descending pathways, classical literature implies that there are four kinds of descending pathways. One from the cortex, and the cortex is involved in the planning and initiation of movement, depending on the kind of sensory stimulus that it has. The red nucleus, so the cortex to spinal pathway is called the corticospinal pathway. It's in the name. The red nucleus also helps in movement coordination. It's called the rubrospinal pathway. The reticular formation, which is involved in deciding the speed, the kind of posture, the gait, and so on. And then the vestibular nuclei, which helps in maintaining balance and coordinating different parts of the body. So these basically send different kinds of input into the spinal cord and so into the motor neurons with different forms of information that all come together to make specific kinds of movements. And so what classical literature doesn't really tell us is what the essence of my talk is about cerebellar spinal neurons. But before I come to that, let's talk about the cerebellum for a while. Now, from back in school, we know that the cerebellum is crucial for motor behavior. We know that it coordinates movements, it maintains balance, it's involved in learning of motor activity, error correction, and so on. And so it makes sense that the cerebellum is a very important cornerstone in the field of neuroscience that actually studies movements. But to a certain extent, the connections between the cerebellum and the spine have been classically ignored, and that's what I'll be talking about later. Now, over and above understanding how movements occur, there is more to why we should study this field. Now normal movement is obviously an incentive to study how these things integrate and conduct behavior. But beyond that, there is also disease and there is also disabilities that might hopefully be cured through exhaustive research on how the spinal cord and the brain and sensory neurons and motor neurons all come together to elicit movement, because there are hundreds of thousands of cases of spinal injuries, of paralysis, and so on that need rehabilitation, but the rehabilitation stops at getting a wheelchair or getting a walker. To this day, there are no real cures to spinal injury. And this was exhibited by Mark Pollock in Trinity, in a science exhibition at Trinity College in Dublin. In 2014, the exhibition was called Fail Better, where Pollock basically exhibited Christopher Reeves, who is famous for being the Superman, back in the movies back in the day. And Christopher Reeve suffered from paralysis, and so did Mark Pollock, who fell out of a window and injured his spine, and so was paralyzed for life, became a quadriplegic. And the point of this exhibition was to demonstrate the importance of studying how movements occur, because there are so many people who can't actually do it. There are so many people who are physically disabled who can't elicit movements that most people in this talk probably take for granted. And that is the reason I added this slide, is just to highlight the importance of studying movement from different perspectives in neuroscience and understanding how these things are integrated. So coming back to descending pathways, I spoke about the different kinds, the four different descending pathways. Now we understand these four descending pathways to different levels of complexity. For example, the corticospinal pathway has been studied extensively. We know the different regions that it terminates in. We know the different kinds of motor commands that it sends. There's been extensive work on basically what the corticospinal pathway is doing. Even the rubrospinal pathway has been studied. So have these pathways to different levels of extent, to different levels of complexity. But as I'll be touching upon again later, there is still a lot of debate about how these pathways function. And beyond that, there are still a lot of unknowns about what else is there. Now we know that these four pathways exist, but are these the only four pathways, or are there more pathways that are actually very crucial to movement that we haven't found out about yet? And now that we know that there are descending pathways, the next question is, how do we characterize descending pathways? How do we actually trace them out? How do we see where they terminate, where they originate, and what different parts of the brain and the spinal cord do they pass through? And maybe like what are the collaterals, what inputs do they get from other parts of the brain and spinal cord? And so there have been a lot of ways to study descending pathways. Through classical literature, there are ways that include accent degradation, where there's people actually go and injure neuronal pathways to uh, facilitate axonal degradation, and then they look at these pathways and see where the degraded axons terminate, and so from there they come up with where these pathways terminate and so on. And now in contemporary, over the last decade or so, there has been a much more reliable way of actually tracing these things, and that is what Anu uses in her paper. She uses a technique called viral tracing, which is basically injecting a virus into a neuron, and this virus actually goes, so in this virus goes to the cell body and replicates, and then you in this virus you add different constructs, you add fluorescent proteins, you add different other elements that help in studying movement, and through that you get a very robust way of actually going into a neuron and studying where the neuron originates and terminates and where else it's going and so on. And so in her paper, Anu uses a very well-established AAV retrovirus. Now, usually AAV viruses are adeno-associated viruses, and these are in today's world very frequently used to genetically target neurons in the brain. These infect the soma, that is this part. Usually what adenosine-associated viruses do is infect the soma and then go to the axon terminals, where the other parts, the other elements in the viral construct, such as fluorescent protein, for example, is expressed, and so you know where the axon is. But what Anu did was use a retro AAV2 virus, that starts at the axon and goes all the way up to the cell body. So now you can start at where this neuron is terminating and see where the neuron originates. So for example, a descending neuron that starts at some part of the brain, say the motor cortex, and comes down to some part of the brain, say a segment of the cervical, a section of the cervical segment of the spine. You can now inject a retrovirus here, and this goes all the way up to the cell body, which is, if these two things are connected, will be in the motor cortex. And then if there's an element of fluorescent protein, the fluorescent protein will go and get expressed here. So now you know exactly where the origin for this axon terminal in the brain is. And she used the RAV2, which is developed in the Kharkova lab, and this allowed the infection of the axon terminal, and it then traveled all the way to the cell body and expressed in Anu's work fluorescent proteins that actually helped her characterize where the cerebellospinal neurons originate and terminate. So now going to exactly what Anu did, she used reporter mice, which they were reporters for TD tomato. And what this is, so these mice have a gene sequence basically, and this is a very broad analysis of what's going on. So it has a fluorescent protein which is controlled by a CAG promoter, and between these two things there's a stop codon which prevents generalized expression of TD tomato in the normal reporter mouse. But this entire feature is tree dependent. And what this means is it uses a tree lock system. Some of you may have read about tree lock systems. What this does is this stop codon is flanked by two lock sites. And then when you inject tree, as Anu did here using the retro AV2 virus, which when you inject the virus with the Cree construct here, the Cree will be transported all the way up to the brain region. And then the Cree is a recombinase, which will, depending on how you position these logs genes, will lead to the deletion of the stop codon, which then enables the expression of TD tomato because the stop codon is deleted and the CAG promoter is in direct connection with the TD tomato. And there is expression of TD tomato in the brain regions that the virus has infected through the neuron. Now, she differentiated the spinal, she injected different parts of the spinal cord, as you'll see through the talk. And it's important to look at different parts of the spinal cord because the spinal cord has gradients of functionality. So the cervical region, for example, controls movement in the neck and forelimbs. Then there's the thoracic region that controls movements of the trunk. And then there's the lumbar regions that control movements of hind limbs. So if, for example, you want to study how hind limb movements are elicited, you can't study the cervical segments because there's a completely different region of the spinal cord that's working on these things. And so when she actually injected Cree into the spinal cord segments and then which facilitated the expression of TD tomato, she found, as she expected, that there were retro-labeled neurons in the motor cortex, which makes sense because the motor cortex is very important in movement, as we know. Then the red nucleus, again, another one of the sites of descending pathways that I spoke about. The vestibular and the reticular formation, again, something that I've touched upon. But what was really interesting was that she also found sites of origins for descending pathway neurons here in the deep cerebellar nuclei. Now, this is really interesting because so okay, before I talk about that, she found, not only did she find origins of descending neurons from the cervical region, but she also found it from the thoracic region and from the lumbar region. Now, this is important because while there are some mentions of the existence of neurons that connect directly from the cerebellum to the spine, classical literature and classical textbooks don't really implicate a direct connection between the cerebellum and the spinal cord. And this now, why is this important? Okay, so there's a connection between the cerebellum and the spine, but why is this important? Why do we care about the cerebellum? Now, we know for hundreds of years that the cerebellum is necessary for motor coordination. You can still move without the cerebellum, but you can't coordinate your movements. And this is because different parts of the cerebellum, different elements in the cerebellum, such as the granule neurons or the deep cerebellar nuclei, indirectly receive sensory information from various modalities that I touched upon, such as touch and proprioception and so on. And this allows the cerebellum to actually gather sensory information and tell a lot of things about how the body is placed in like 3D space, or how fast the body is going, if say a body is moving, how fast it's moving, and so on. And not only does it get sensory information, it also receives commands of the motor copies from other parts of the brain, such as the motor cortex. And this actually gives the cerebellum a sense of what should be happening. So suppose we are walking and we are walking at a certain speed. Now there is sensory information about the speed that we are walking at. But say that the motor commands sent by the brain require the body to walk at a certain different speed. Now, what the cerebellum can do, because it receives both sensory information and copies of the motor command, is that it can actually predict errors in movement. Errors in this context are differences between what the motor command that the brain sent to motor neurons to elicit a certain movement actually require the body to do, and information from the sensory modalities that is saying what the body is actually doing. Now, often these might be the same thing, in which case there is no error, and there is no error between the motor copy and sensory information. But sometimes the you might want to have done something when you're reaching, for example. You might, if you're reaching for a bottle, in the normal case, you reach for a bottle and there is no error, it's fine. But say you miss the bottle, say you reach for it, but you missed it. Now the brain sent a motor command that gave the location of the bottle in 3D space and a command of how to actually reach it. But then real-time sensory information then tells you that you actually missed the bottle. So the error signal that is generated can actually be processed by the cerebellum. And this actually makes it a very important region to study in motor behavior, which actually makes it surprising that for the longest time we thought that the cerebellum does not have direct access to the spinal cord. Beyond this, a lot of our movements happen in the order of milliseconds. They happen very, very fast. And the cerebellum evidently seems to be this major conduit in actually facilitating behavior. So it's surprising that for the last 100 years, we've not found, apart from mentions here and there, we've not found direct access between the cerebellum and the spinal surface. And just to show the extent to which we actually don't know about a direct connection between the cerebellum and the spine, very, very fundamental textbooks of anatomy and neuroscience, such as Gray's Anatomy. This is a quote from Gray's Anatomy. It actually says that the existence of fibers from the cerebellum to the medulla and the spinal cord is very uncertain. Even the very foundational textbook in neuroscience, Eric Kandel's Principle of Neuroscience, shows a very circuitous route between sensory input, spinal neurons, and how the cerebellum connects back to the spine. There's no direct connection between the cerebellum and the spine. It goes through other regions. It goes through the midbrain, it goes through the brainstem, and so on. So what I want to emphasize is that even though literature has mentioned the existence of cerebellospinal neurons, that is direct access between the cerebellum and the spine, in papers through the years, even very classical textbooks and classical literature actually either denies or says that a direct connection between the cerebellum and the spine is uncertain. And so now what Anu wanted to do was, because there have been mentions of this existence of cerebellospinal neurons, she wanted to establish a framework for actually studying this, or actually proving the existence of a direct link between the cerebellum and the spine, and then actually going beyond that and characterizing them, seeing their organization and characterizing their function, and so on. And so to do that, first she actually looked at how the cerebellospinal neurons are organized in the different deep cerebellar nuclei, because the deep cerebellar nuclei themselves are very anatomically and functionally diverse. There's the interpositus, there's the anterior interpositus, there's the posterior interpositus, there's the vestigial nuclei, and there's the dentate nuclei. Now, each of these different deep cerebellar nuclei have been implicated for different functionalities, such as postural control, for limb control, for Anglican conditioning, and so on. So to start with, it would make sense to have a very distinct grasp on the organization of cerebellospinal neurons within the DCN themselves. And to do that, Anu used the retro AAV2 viruses that I mentioned earlier, and she injected these in two parts of the spinal cord, one in the cervical segment and one in the lumbar segment. And the cervical segment expressed GFP, which is green fluorescent protein, and the lumbar segment expressed MCHD, which expresses red, as you can see here. And so then what the authors found was that the cervical segments went up and were organized contralaterally in interpositors, that is in P, and the vestigial nuclei. And these constituted about 10% of all the nuclei in these DCNs. And she also found that ipsilaterally, so coming back, contralaterally is basically when when the neuron goes from one side to the other. So suppose this is the left side of the spine. When I say contralaterally, I mean that these neurons cross the midline somewhere. It could either be the spine or it could be the brainstem and so on, and go to the right side of the cerebellum. So as can be seen here, this starts at the left side at the cervical and goes to the right side of the cerebellum. And and again, then the lumbar origins. Can you hear me? Because this says that my internet connection is unstable. So,
Yeah, it was unstable for a bit. It's fine now.
oh, okay. So then that neurons that terminate in the lumbar sections or originate in NTA, that is anterior interpositus. And what is even more interesting is that she found certain hints of somatotopy within these different DCN. So as you can see here in the int A, there seems to be a division between where neurons that terminate in the cervical originate and neurons that terminate in the lumbar originate. Zooming into this picture, so this is where the cervical neurons originate, and this is where the lumbar neurons originate. And there seems to be a very distinct difference between where they're located. And if you think about it, this makes sense because the cervical and the lumbar region, as I mentioned, control different parts of the body. The cervical controls the neck and the forelimbs, and the lumbar controls the hind limbs. So it makes sense that there is a functional gradient. It makes sense that there is an anatomical gradient since there exists a functional gradient, which makes the actual finding, the fact that you can see a distinct anatomical gradient, very interesting. Now the authors are at a stage where they've characterized where neurons that connect the cerebellum to the spinal cord regions originate. So they've characterized where different neurons that terminate in the cervical and in the lumbar originate in the different deep cerebellar nuclei that I mentioned. But now the next step is looking at function. So now you have an anatomical tracing of where these neurons are, but what do these neurons do? So to do that, the authors decided to silence the cerebellar spinal neurons that terminate in the cervical regions. And the system that they used for actually silencing these neurons was a three-step strategy where they injected HM4DI. Before I get there, they injected in mice. They injected Cree in the cervical sections, and the injection of Cree was important because in the brain itself, they injected regular AAV, that is AAV that goes from the soma to the axon terminal. And this AAV virus, the AAV virus contained a construct of Cree-dependent HM4DI. And what this basically does is HM4DI is a mutant of a receptor that acetylcholine binds to, and the result of the mutation of this receptor is that HM4DI can also bind to CNO. And this is basically the essence of this three-step silencing strategy. It's that when CNO is then injected in the mice, the neurons that express HM4DI are silenced because the injection of CNO, the binding of CNO to HM4DI obstructs the binding of acetylcholine, which is in the context of the CNS, usually an excitatory neurotransmitter. So basically what happens is the neurons that are usually active here in a normal mouse are silenced, and this confers a level of specificity because not all neurons in the DCN are silenced, or not all neurons in the spinal cord region are silenced. Only the neurons that terminate in the cervical region and originate in the specific DCN are silenced. And this happens because this HM4DI is Cree dependent, and the Cree is only injected in these cervical sections. So that means only neurons that terminate in these cervical sections actually receive this Cree, which means only the neurons that actually terminate there have the Cree dependent HM4DI expression. So now, upon silencing of these neurons, the authors conducted this behavioral assay called the single pellet leaching task. And the single pellet leaching task is basically what it consists of is putting a mouse in a cage. And so this is the cage, and the cage has a slit. And the point is that the mouse reaches for a food pellet that can be seen here. It's called single pellet because it's usually a single pellet on a pedestal, and the point is that the mouse reaches for this pellet and eats it. Now, this is an interesting behavioral essay because reaching is not a natural behavior for a mouse. Usually, when mice go to get food, they use their mouths or their tongues to directly access the food and ingest it. So, reaching is something that the mouse actually has to learn, which means that which makes this a very interesting behavioral assay for actually seeing how motor learning and motor control works. So now the results of actually silencing these neurons were scored based on how successfully these mice could conduct the single pellet reaching task. And so I'll just give you an example of success. So what's happening here is the mouse is reaching, grasping the food pellet and taking it all the way back to its mouth and being able to eat it. An example of failure is the mouse is reaching, but then it dislodges the pellet. It's not actually able to bring the pellet back to its mouth. There are other kinds of failure too, but the essence is there are two results of this. There's successful ingestion of the pellet and there's a failed ingestion of the pellet. So now, because the point is to study how silencing cerebellospinal neurons actually affect motor control. The mice that were used in the assay were divided into two groups. They were divided into mice that had HM4DI, but instead of cyano, there was an injection of saline. Now, saline is not going to silence those neurons because it's not going to bind to HM4DI, and so normal acetylcholine binding is possible. And so, what they found was that the mice, what usually happens is that they start with 30% success or 40% success and then go up as normal wild type mice learn this task. The success rate goes up. But then what they found was that the mice that in which cyano had been injected failed to actually improve through the training period. And so this would be the learning phase. So now this is a very evident indication that the cerebellospinal neurons are important in some aspect of motor control or motor learning. But what aspect is this? Is the mouse actually not being able to learn how to reach itself, or is there a problem in like fine motor control and like moment to moment coordination of how the reaching needs to occur? So to test for this, the authors took the mice in which saline was added instead of cyano and which had been performing well over the 10-day period. And they divided this group of mice into two other groups. One of them continued to have vehicle being injected into them, vehicle saline, and the other group had cyano injected into them. And what they found was that the mice with saline injected into them had similar success rates on day 9 as well as on day 10 and beyond. But the mice in which cyano was injected, which went and bound to HM4DI, there was a distinct drop in performance of these mice between day 9 and day 10. And this implicates that these neurons, these cerebellospinal neurons that are connected ipsilaterally to these DCN are required for online refinement of motor control. So they might be important for the actual learning phase, but even beyond the learning phase, even once the mice have actually learned how to reach, they are still important in actually refining the motor behavior moment by moment with online modes of sensory input. So that's what's going on in the cerebellospinal neurons that are connected ipsilaterally to the DCN. But then there's, as I mentioned earlier, there's also another group of cerebellospinal neurons that are connected contralaterally between the cervical and the DCN. So what do these neurons do? Now, it has been found that the interpositusity and the vestigial, they are important for postural instability and ataxia and actually coordinating movements and so on. So what the authors wanted to find was that are these neurons actually important in motor performance again? And if so, in what way? So they used a behavioral essay called rotor rod, where there's basically a rod that accelerates and the mouse has to balance on this rod. And the balance requires that the mouse coordinates different parts of its body, so it has a constant, um, it can constantly balance on the rod throughout the behavioral assay. And so over here, what the authors did was the same injections as I mentioned last time, except this time they used M-cherry. They used a double flux HM40I system. And what they found was that the mice that had injections of cyano went as good in the training period as the mice that had injections of saline. And so now this is again an indicator that even the cerebellospinal neurons that are contralateral, that have a contralateral connection between the cervical and the DCN, are important for motor control and body coordination. So now that they knew that these nuclei were important for this, for motor control, the next step is to actually go and see exactly where in the spinal cord do these neurons terminate. Because up till now, I've been saying it's the cervical region or the lumbar region, but the cervical region itself has multiple sections. There's C1, C2, and so on, and the lumbar region also has multiple sections. So it's important to know that exactly where, exactly in which section is it C1, it is C5, and C6, and so on. And within this section itself, there's the there's different gradients of functionalities of these neurons. So each section of the spinal cord is divided into laminae, and each laminae has been implicated in different functions. For example, laminae 1, which is towards the top, and laminae 2 have been implicated in getting sensory input and getting inputs of pain and sending inputs of pain to the brain. And then as you go further downwards towards the end of it, proprioceptive input is received in these regions and then sent upwards or downwards to the spinal cord. So basically, even in each section of the spinal cord, there's a distinct anatomical organization of neurons with different functions. So what the authors wanted to do was see exactly where the contralateral cerebellospinal neurons actually terminate. So to do this, what they did was they expressed a fluorescent protein in the DCN, in the contralateral cerebellospinal neurons. And they did this by injecting Cre again. And this is a double law system. So when Cre is injected, the neuron terminals over here will light up, expressing red. Because there's M-cherry, they'll express red at the points where these neurons come and terminate. And so what they found was that there, in the brain region itself, they originated from the INPI and the vestigial regions. That's what you see here. And if you trace how the neuron is actually organized downwards between the cerebellum and the spinal cord, the neurons actually hook contralaterally into the spine. So that is the organization downwards from the DCN, and then they travel through different parts of the brain and the spinal cord, and they terminate onto lamina 8 significantly, and also into lamina 7, which are very major hotspots for pre-motor neurons. That is, neurons that go and synapse onto motor neurons. So that means these neurons actually control how motor neurons work. So it's interesting now that these cerebellospinal neurons come and terminate into these regions, because these regions, as I mentioned earlier, these laminae are directly involved in the actual control of motor neurons. And digging deeper into this, uh, they, uh, they actually found different cell types that these cerebellospinal neurons connect to. And so why is this important? We've already gotten here where we know which lamina of the spinal cord section these cerebellospinal neurons terminate into. It's important because while there are functional distinctions between the lamina, we've gone through that, there are actually many types of neuron types that are present, and cell types that are present in the spinal cord and the brain. And each of these neurons have different morphologies, have different functions, have different neurons that they talk to, different regions that they actually target. So it's for a very clear understanding of exactly what the role of these cerebellospinal neurons are. It's important to actually dig beyond just the anatomical organization of these neurons, and go and look at what types of cells are these neurons terminating into. So the first type of neuron that was the interesting that these neurons terminated to were engrailed 1 neurons. Engrailed 1 neurons are basically a class of inhibitory neurons that regulate locomotor speed by controlling the activation of motor neurons. And what they found was that about 30% of the engrailed 1 neurons received input from the contralateral cerebellospinal neurons in lamina 8. Now, how they did this was kind of similar to the retrovirus strategy that they used before, except over here, what they had was reporter mice that reported TD tomato, and what they did was these mice also had tree-dependent engrailed neurons, and so when and then sorry. Over here, they found that basically what they found was that as they traced the cerebellospinal neurons to lamina 8, where the engrailed neurons also reside, they found by viral tracing that the cerebellospinal neurons synapse into where the engrailed 1 neurons actually lit up. The second type, so now this is in the cervical region of the spinal cord, right? These are inhibitory neurons in the cervical region of the spinal cord. But we've also discussed how the cerebellospinal neurons seem to be important for whole body coordination, as we saw during the rotor rod behavioral essay. So now, how is whole body coordination actually accomplished? And this makes the next discovery very interesting. The authors also found that cerebellospinal contralateral neurons synapse onto long descending propriospinal neurons. And what propriospinal neurons do are propriospinal neurons connect different sections, different regions of the spinal cord. So they conduct information from, say, the cervical region down to the thoracic region and to the lumbar region, which means that information and control in the cervical region is also translated into the thoracic and the lumbar regions, which kind of implies that there these are important for the coordination of different parts of the body simultaneously. So over here, what they found was that about 45% of the propriospinal neurons had synaptic inputs from the cerebellospinal contralateral neurons. And again, they used viral strategies similar to the ones we have discussed before. And so I've spoken about the propriospinal neurons and what they do. And then the final type of neurons that these cerebellospinal neurons also synapse to are commissural neurons. Now, commissural neurons are types of neurons that connect one side of the spinal cord to the other side of the spinal cord. Now, this makes it evident that these neurons are important in left side and right side coordination. So in movement between the left and right side of the body. So it's very interesting that these cerebellospinal neurons actually also target these commissural neurons. So that means that not over here you can see in lamina 8, another paper, Zhang et al. In 2013 showed commissural neurons originating in lamina 8 and lamina 7, which is where, as I discussed before, cerebellospinal neurons terminate. And so to sum up, what we've seen, what the authors saw, was that cerebellospinal contralateral neurons, the cervical contralateral neurons, synapse onto both segmental neurons and intersegmental neurons, which enable coordination both within a spinal cord segment and throughout the spinal cord through propriospinal inputs. And these also connected to different types of neurons. They connected to inhibitory neurons that have specific roles. That, for example, in grade 1 has a specific inhibitory role in regulating activity that finally translates into how motor behavior is elicited. So now we've gotten to how to where the cerebellospinal neurons originate and where the cerebellospinal neurons terminate, and what classes of neurons or what types of neurons do these cerebellospinal neurons synapse to. And now we've got a sense of what their role might be in actually coordinating movement through the body. But it is also important to realize that these neurons not only go to the spinal cord, but they also go to different parts of the brain. There is heavy generally through the brain, there is heavy collateralization of neurons, of descending neurons. So not only do these cerebellospinal neurons go to the spine, they also go to places such as the thalamus, which is also known to project to the motor cortex, which makes it important in the study of behavior. It also goes to the red nucleus. These neurons actually branch out and terminate to the red nucleus. They also terminate to regions of the pontine reticular formation and to the vestibular nucleus and the reticular formation. Now, if you actually look at the names I've been dropping of the regions in the brain that these cerebellospinal neurons target, you will find that I've mentioned these before. The motor cortex, for example, had its own descending pathway. The vestibular nucleus and the reticular formation had their own descending pathway. The red nucleus had their own descending pathway. This implies, so again, those descending pathways are shown, and schematics of where they terminate are shown. So what this implies is that not only do cerebellospinal neurons have direct control over regions that they terminate to, they might also, since they branch out into regions that have their own sets of descending neurons to regions in the spinal cord, these neurons may also have control over how these descending neurons send information to the spinal cord to elicit motor behavior. So what kind of control might this be? What are the significance of these pathways and how they are correlated do these pathways send internal copies of the commands they're sending to the spinal cords between themselves? These are open questions in the field now that several people are working on, including us. And hopefully through research, we'll actually find how this integrated circuitry of descending pathways actually come together to facilitate the very intricate motor behavior that we express every day without actually consciously thinking about it. And so since the authors did ask this question, another more specific question of control of how cerebellospinal neurons control these other descending pathways could be that do the cerebellospinal neurons actually regulate the timing of how the motor commands from the different descending pathways are sent to the spine. That means, do they control the timing of the activity of the neurons that connect these different regions of the brain to the spine? That might be possible. On the other hand, there could be something completely different going on that we have yet to study. So the reason I brought up whether the cerebellospinal neurons actually may play a role in regulating the timing of the activity is because it has been shown before that when the in a decerebellated animal, that is an animal missing the cerebellum, recordings from the vestibulospinal neurons during locomotion showed that compared to the controls, where there is a rhythmic oscillation of activity, decerebellated animals had constant activity, and their activity did not change over time. So, which implies that there is a possibility that the neurons that connect the cerebellum to these places, a subset of which are the cerebellospinal neurons, might actually be involved in regulating the activity of these other descending neurons too. So that is an interesting question to ask, that how these networks of descending pathways are actually integrated and how they all come together to facilitate movement. So another interesting question that remains unsolved is whether these neurons, the cerebellospinal neurons, actually receive input back from the same regions that they terminate onto. That is, so say if int A terminates into a section of the cervical neuron, do these brain regions also get information back from the same parts of the spinal cord? And if they do, this might actually be a way of facilitating rapid error correction that we've spoken about before. So these are all interesting questions that hopefully will be answered in the coming years through more extensive study on how motor behavior actually takes place through the integration of these different elements of the body that participate in motor behavior, such as the spine, the cerebellum, the motor cortex, and so on. So to summarize, what the essence of the talk was that there is distinct proof of cerebellospinal neurons, and furthermore, these cerebellospinal neurons have been anatomically, their anatomical organization has been characterized through ipsilateral and contralateral divisions between the various DCN and different sections in the spinal cord. And so overall, excitatory neurons from the int regions and the fastigial nuclei target distinct segments in distinct halves of the spinal cord. So the ones that are ipsilateral are important for skillful limb control, as we saw from the results of the reaching task. And the ones that are contralateral are important for learning complex locomotory tasks, as we saw from the rotor rod essay. And not only do these cerebellospinal neurons have direct influence over the regions they terminate to. They have also been found to connect to different parts of the brain. The same neurons connect to different parts of the brain, which implies that there might be some level of control that these neurons have upon other descending pathways that go down from the brain to the spinal cord. Yeah, so basically what we saw was that there's intersegmental and segmental control of these descending cerebellospinal neurons within the body of the mouse. And these are so because of the segmental and intersegmental control. Not only are these important for specific regions of the body, such as forelimbs and hind limbs, they also help in whole body movement coordination. So that that brings us to how the authors of this paper prove the existence and characterize a fifth descending pathway apart from the ones that I mentioned before. That is the cerebellospinal pathway. And I'll end my talk here. Thank you so much. Are there questions? If there are questions, please feel free to hop in and ask. I don't think there are any questions on the chat. So please feel free to open your mics and ask.
It was very articulate. I almost understood everything. But so my question is, what exactly is that Cree-dependent reporter? I would like to know about it.
Okay, so the Cree log system is very broadly, the Cree is a recombinase. And so you know what recombinases do, right? Recombinases can cut different parts of the DNA and transfer them from one location in the DNA or gene to another. So what usually happens in a tree log system, I'll give a very specific example. Say you want to silence a gene through deletion. What you do is you would flank gene X with two different log sites. Logs are basically specific DNA segments with specific properties. So these two log sites will be used to flank this gene of interest. And depending on the direction in which the log sites are, so if the log sites are both directed from 5' to 3', what happens when the Cree recombinase comes to the segment of DNA? It recombines this DNA such that the gene segment within the log sites are deleted. So basically, that is one way of using the log sites. Another way of using the log sites is for gene inversion. So if you invert how the log sites are with respect to each other, you can actually facilitate the inversion of a gene between these two logs. So basically, that's what the Cree log system is. It's a system to manipulate DNA segments for in this case, for example, to study neuronal silencing, but it's been used throughout for gene expression. It's been used to actually it's been used to add different things like channel reductions and all to neurons by actually going inside and manipulating the DNA and so on. So it's a way of manipulating DNA.
Okay. So basically, those flanks, they are homologous kind of thing that.
Yeah, they are homologous flanks. They're the same. They are log species. They're the same DNA segments with the exact same characteristics. It's just that Cree recognizes these log species and comes to these sites and carries out its recombination. And depending on how these log species are oriented and how many log species are there. If you remember, I mentioned that in one of the slides, I mentioned that a certain fluorescent protein was double floxed. That means there were two sets of flox sites. If you remember, there was DIO, written in one of the slides. I'll have to go back a long way, but I can send you this presentation. But so DIO is basically double floxing, where two sets of lox-based sites are flanking a gene of interest. And so through permutation and combination, you can manipulate the DNA in different ways using 3D that recognizes lox-based sites.
Yes, Shubham, right.
Yes.
So for example, in flies, you also use inhibitory RNAs, right? So
Yes.
those don't work very well in mice. So in mice, Prelox is more popular to delete genes. So it's kind of, you could think of it that way, but it's a slightly different mechanism. Yeah.
These things always fascinate me, like how things have been made to, facilitate our.
Even things like viral tracing, these are relatively new. I mean, back in the day, neurons would be injured and through degradation, they'd study how neurons connect to other parts. And then things like lesioning studies or full decapitations would be done to see how like motor behavior changes. But now there are far more both subtle and robust ways of actually manipulating different neurons and seeing how they behave. And they are just becoming more and more efficient. So hopefully, through in the coming years, we can go leaps and bounds in how we study motor behavior. And it's not only motor; it's in different aspects of neuroscience and biology. So it's an exciting time to be in these fields.
Okay, thank you. If anyone else here wants to ask question, you can go ahead. If no one else is interested, Anu, so what kind of research do you do? Like, do you use mice model system to do tracing and all?
Yeah, so we use a lot of, I mean, we mostly work on mice. That's our model system. So most of the tools that we talked about, like, so we do a lot of viral tracing to find out how different parts of the brain and the spinal cord are connected to each other. So these viral tools, like Shrivas mentioned, they offer like much, finer interrogation of like these neural circuits. Before, people just used to do degeneration studies and things like that, but now you can actually see even connections between different parts using these viruses. So we do that, and then we our basic strategy or approach is to silence or activate neurons in different parts of the brain, specific circuits, and then look at what effect it has on behavior. So, and when I say behavior, it could be a lot of different things. For the most part, we are interested in setting up like behavior assays for assessing like limb control, like how, for example, like how do you pick up a coffee mug? Like it's something that you learn as a toddler, but then, so single pellet reaching assay is one of the things. So we are trying to take that to the next level by actually being able to score the outcomes. For example, rather than just say success or failure, we want to kind of really say, why is it a failure? Like, why did the mouse fail? The other thing that, and we're also interested in how the nervous system processes pain information. So we do that in collaboration with another lab here. So.
Interesting.
Yep, and yeah.
So Ayush, it's
Yeah, so I don't think anyone is going to ask any more questions. So
Yeah.
thank you for the talk. So,
Yeah.
Thank you so much for having me. Thanks for your time.
Thank you so much. This was fun. It was nice interacting with all of you. Yeah.
thank you.
Thanks, Shiva.
Thanks so much.
have a good evening.
You too. Thank you.
And we can discuss what you're doing.
Go ahead. Hey, so Shubham, what do you work on?
Oh, so I am working on a tracheal system of flies. So trachea is a tube, a breathing tube of flies. So we study how certain development, like cell development happen, like morphogenesis in cell happens. We try to, uh, fill in, like we try to understand the whole picture of cell development during the metamorphosis from different stages, like from larval to pupal transition, or from embryo to larval transitions. And the project right now I'm working on is a cell which differentiates itself into a very branched form, and because it needs to branch in order to transport oxygen from trachea to different tissues. So,
Okay. Oh,
so,
okay.
so I'm trying to understand what causes that branching, like what events inside the cell lead to those branching. So I'm trying to build a story right now to understand.
Okay. Cool.
We use a GAL4 system to
Base system. Yeah.
yeah,
Drosophila
use,
genetics is way more advanced than mouse genetics. We
yeah.
always.
So we
Yeah.
do different kinds of silencing, knockdown, and see
Okay.
fluorescence. Yeah.
Okay. And what are is there a specific pathway you're going after?
So I'm basically looking at the cytoskeleton aspect of it. Like cytoskeletons, they help in branching. So I'm tracking how, what exactly is happening, which cytoskeletons, like tubulin, actin, myosin. I'm looking at those things, and I'm trying to see like there is an element of force as well. Like the forces are working there to drag the cell membrane from.
Okay.
So there are a lot of things that like I have just recently started. Like I'm working in Professor Ranjit's lab
Okay.
from my second year, but I was not working on project. I was just learning to do things, and now I'm starting project since I have learned the basic things. So that's my project.
Okay.
That's good.
Yeah.
So we do have a neurobiology course here. Like last semester, I did my neurobiology course, and we studied different things, but of course we didn't get to study too much. Like it was just a 3-4 months course. So,
Okay.
but
Well,
we
when I
did.
was.
We did some of the papers, like some research papers, like our professor who did in his PhD and postdocs, I guess. So we did do those papers, like we studied how he did those tracing and all, but we didn't do that Cree thing. So I was interested about it.
Yeah. Basically, it's also a nice time to be doing neuroscience in India, because usually like last 10 years there's been so many more labs that have started doing neuroscience compared to like 10 years ago. Um, yeah.
I was interested more, like I was interested in worms, like especially how RNAs work, in different aspects of inheritance, but we don't have a lab here. So so Professor Ranjit kind of taught us in our first year, and he's also very good speaker. Like he's really good speaker. So I got I was mesmerized by his speaking, and I just after finishing my first year, I went to him and I asked him, sir, can I do some project with you? And then I just never left him, and I'm stuck here, kind of.
There's a lot of really good C. elegans out there.
Yeah, I think there is a C. elegans lab in IISc. I'm not sure.
There is one, yeah.
Kavita Babu, so Kavita moved from ISA Mohali so she works on C. elegans, and they do really, they work on the neuromuscular junction, so they do pretty cool stuff. There are no Drosophila labs here though.
So Shrivas, are you planning to do masters? Are you in 4th or 5th year?
I'm doing an integrated master's. I'm in my 4th year. This is my 5th year. So I'm actually in this lab to do my master's thesis. That's what I want.
Okay. So you'll do PhD, or you are planning something
I'm
else?
planning to stick on in neuroscience. So that's the next year.
Stuck. Yeah.
Which year are you in, Shubham?
I'm in 3rd year,
Yeah.
6th semester.
So your integrated program will have 5 years, right?
Yes.
Okay, so you got time to settle down and do neuroscience.
What?
You got time to actually explore neuroscience now and settle down and make a video.
To be honest, I love all kinds of biology.
Yeah.
In general, everything. Like I just like making, like building stories. Like just like you just mentioned how from starting from finding one thing to another. I just like that process. So it's not like I'm interested into one thing. It's like,
That's great.
Yeah.
okay.
Yeah.
So are you all right talking? So since I've been out.
No, I just said that's the most fun part, like building those stories. So yeah. Okay, I need to run, but yeah, thank you so much for having us.
Yeah.
It was really fun.
I'll keep in touch with some of the doctor. Ayush, I think, who reached out to us. So
Yeah.
I'll keep in touch. All right. Thank you so much.
Thanks, Ayush. Yeah.
Good evening.
Thank you for joining us. It was fun. Yeah.